Good evening, everybody, and welcome to this IF Oxford event tonight called NeuroTales. It's going to be a storytelling-based event, and hopefully you'll find out some really interesting facts about people's lives and the work that they do and why. It's a pleasure to pass on to the host for tonight, Corinne Piekma from the University of Oxford, and she's going to guide you through the NeuroTales for this evening. Corinne, over to you.
Thanks very much, Dane. Hello, everybody, and welcome to NeuroTales. My name is Corinne Piekma, and I'm your host this evening. The theme of tonight is happenstance, the small chance encounters that can have a huge impact on the paths that we take. And it was those chance encounters that set our three storytellers of tonight out on their career paths into science. Each one of them is now trying to understand something about how our brain works, but it's the story of how they got here in the first place that we are telling tonight. We often think about scientists that, you are born a scientist and then after school you go to university, you do your PhD and possibly a couple of postdocs, and then you go on to a professorship, hopefully in a nice, neat, straight line. But if we scratch beneath the surface, it turns out that there are actually very many different routes in which we can become scientists. Our first storyteller, Jesper Anderson, thought that he was destined to work in a factory, but things have turned out quite differently for him. Before I hand over to Jesper, I just want to say, to repeat what Dane said, we have some time for questions at the end of each speaker, so please pop them in the Q&A box if you have any. And now, without further ado, here is Jesper's NeuroTale.
Thank you very much, Corinne, and hello. My name is Jesper Anderson, and I work at the WIN, the Wellcome Center for Interactive Neuroimaging, essentially a place where we take pictures of the brain. And as Corinne has said, I'm going to talk a little bit about my path to academia, because it's quite circuitous and what have you, and also a little bit about two people that I met on that path. Also, what I also first want to say is that I don't actually do research about the brain. My research aims at improving the tools that are available for people that do research about the brain. So to make this very, very concrete, so let's say that your research involves taking pictures of the brain with a big scanner, an MR scanner, for example, then someone who works with MR scanners to make sure that we get the best possible images from that MR scanner would be someone like me, a methods researcher. And these methods researchers, we tend to be engineers, mathematicians, physicists, or some such thing. So I am Swedish. I come from a very, very small town in Sweden, which you have probably never heard of. And I would say that I'm probably the most unlikely academic, or I started out as the most unlikely academic you could ever imagine. I come from a very, completely non-academic family. And in this little town, we had a couple of factories, and everyone I knew worked in, or parents worked in one of those factories. So they just assumed that I would too, and that's also what I did every summer, so growing up. So yeah, I took that for granted more or less. But as it turned out, I did quite well in school, and at some stage I decided, well, maybe I should try and go to university, which wasn't a big deal, but. At the age of 18, when I left school, I went to a place called Uppsala. And Uppsala is a lot like Oxford. It's a sort of smallish city, smallish town, quite dominated by a big university, but there's a little bit of town attached to it as well. And there I studied engineering and physics, still very much, with the intention of going into industry, working in a factory, probably still, but maybe, not on the factory floor, but rather maybe doing something else. I did my physics studies, and after I'd done my studies, I got a job at a place called Scannetronics. And Scannetronics, again, is a small, well, mid-sized company, maybe a couple of hundred people in total, in Uppsala, the same place where I went to university. And what they did was they built cyclotrons. And what a cyclotron is, that is essentially a machine that produces radiation. And these machines that produce radiation, you can either use them to radiate cancer patients and try and cure cancer that way, or you can irradiate little samples and make them radioactive. And then you can then inject those radioactive samples into people, either for treatment, but mostly for diagnostics, and sort of pick up where that radiation comes from with a scanner, like a PET scanner or a gamma camera, something like that. It was very much an engineer-driven company. It was a nice company. And at some stage, someone in that company decided, well, we're building these cyclotrons. So the people who are buying our cyclotrons, they're probably buying our cyclotrons because they want to have a PET center, or they want to produce radionuclides for PET, positron emission tomography. And what positron emission tomography is, so you're probably all more familiar with magnetic tomography, or MR, which is what PET was, or positron emission tomography was. That was sort of the predecessor of MR, in the sense that in order to get the pictures that we took, you need to inject radioactivity in people first. So it's a little bit damaging in that respect. So MR is nicer. This was doing at the time. So anyway, someone at this company decided, well, we are building these cyclotrons. Wouldn't it be good if we could also sell a PET scanner to these customers? And because everyone in this company were a cyclotron engineer or something, this was all a little bit sort of stepmotherly treated. And what they did was they hired 10, 15 very, very young engineers straight out of university to start building these PET scanners. And which seems quite astonishing that we actually managed to do that. So 10, 15 engineers, we were all in our 20s. I was probably the most junior of us all, and we were building what was probably the best PET scanners in the world at the time. And this was all down to one, well, I wouldn't say all, so that would be insulting to some of the other people in the company, but it was this one person in the company, let's call him Sven. That's a good Swedish name. And Sven was a genius. In my career, I would say that I met 3 people I would call geniuses, and Sven was the first one. And he was, without him, there's absolutely no way that this group of inexperienced young engineers could have built these scanners. My first meeting with Sven was. So I think I was probably the last person employed in the company. So the others had been there for a year or 2 already, and I was down in the assembly hall, and I was struggling a little bit with a software, so a computer program I'd written to run a radiation detector that I was experimenting with. And I couldn't get it to work, and I see this guy Sven come into this little room where I was working. And I knew who he was, but I hadn't actually been properly introduced to him. So he came in there. It was very clear that he was busy and he didn't want to look at me because I might ask him something. But I was like, okay, I'm struggling here. So I asked him that. I said, well, I'm really struggling with this program here. Could you please come and have a look? He was okay. I came, looked over my shoulder to the little screen, and the screen back in those days, screens were little, looked over my shoulder at the code I've been struggling with to try to get to work. And he says, well, there's an equal sign there. That should be equal, equal. Okay, and he was right. And then he looked at me and he said, well, you're not very clever, are you? And it was said without any malice or anything like that. It was just useful for him to kind of know roughly where I was. So he didn't give me any too hard things to do. But Sven was absolutely amazing. He was a complete genius. And I wish, we were living the dream, the dream of an engineer. And I wish we could have gone on to do that. But what had happened was that the PET division of this company was purchased by General Electric. And what they really wanted to do, they wanted to sort of buy the brains who were building these scans. In particular, they wanted to get Sven's brain to go to Milwaukee at the General Electric there and keep building PET scanners. He had no interest whatsoever in that, and they pretty much didn't get anything out of this. But what happened for us who worked there was that we no longer had a place where we could build PET scanners. We all needed new jobs, and we scattered in various directions. And what happened to me and one other guy from this company was that in this little town, Uppsala, there was a pet center that had recently bought 2-hour scanners. And obviously they thought, well, this is fantastic. We can now hire 2 of the people who built these scanners. And that would be, they can keep our scanners going and they can do all sorts of other things. So that's what they did. So I now started working in an academic institution, a pet center, but still very much in a non-academic role. I was a research engineer. I was responsible for making sure that the scanners were running and that kind of thing. And we also started building various things. We started building a robot for taking blood samples and measuring the radiation in blood samples, things like that. We did much the same things that we did at the factory, me and this other guy that they hired, but on a smaller scale, because obviously now we didn't have a huge big assembly hall and things like that. But we tinkered, we solved problems, any problems with the data, with the scanners, with the blood sampling systems, we solved. And that was it. I was an engineer still. But then at some stage we had a visitor come and look at the pet center. He was an academic. I think he might have been from Britain even. And we showed him around and we showed him the things we've done and the various problems we had solved. And he said, well, actually, we all have these problems. I mean, these are not your problems. Every pet center has these problems. You could probably publish this. Okay. Obviously, I knew by then what scientific publishing was, because our doctors and what have you, they published papers. I had never thought that I could do that. But what I did was I published a few papers. Turned out he was right. These problems I had solved, they were of a general nature, so I could publish this in scientific journals. And in Sweden, when you write a PhD thesis, it's different to in the UK. In the UK, you actually properly sit down and write a thesis. In Sweden, what you do is you publish a bunch of papers. And then once you have 4 or 5 or 6 papers, you just staple them together and you call it a thesis. So that's what I did. So now suddenly I was an academic. I had a PhD without really at any stage thinking that I would do that. Now, Sven was the first person I wanted to talk about. Now, I want to talk about the second person, and let's call him Bengt. Again, it's a very good Swedish name. Now, Bengt, he was my boss at the pet center. Bengt was a northerner. Northerners, much like the English northerners, very down-to-earth people, maybe Swedish northerners even more so, because it's just further north. He was this elite basketball player, 6'5", went to the US after his PhD, came back, no longer a 6'5" elite basketball player. He'd expanded his mind in the US. He'd also expanded himself in the US, and he came back as a 6'5" diameter cannonball. And cannonball, that was, that's the way he approached life. And he just, went through life like a big cannonball. So if you can imagine Steve Jobs, only from Newcastle, weighing 450 pounds and having absolutely no dress sense at all, that was Bengt. And he was by far the most entrepreneurial person I've ever come across, and that I probably ever will come across. He was incredibly impressive, and he was a powerhouse of a person. I'm going to tell you a quick story that just illustrates a little bit who he was. He also had he was very temperamental as well. And there was this one time, and we had visitors to the pet center, and he'd been to the bathroom. And in the bathroom, he discovered that someone, and he assumed it would be one of us working there, had also been to the bathroom and hadn't cleaned out the bowl after him or herself. And he got absolutely furious. He called everyone in. He had no idea what was going on. He called everyone in to the little conference room. And then he stood in front of us all for 10, 15 minutes, telling us all. These were, 2 professors, a bunch of assistant professors, associate professors, a bunch of PhD, sitting there, being told by this huge guy walking around there, telling us the importance of cleaning out the bowl after you've been to the toilet. underlining every word he said with a toilet brush. I'm not kidding you. The toilet brush in his hand. He was waving around like this, and it was, you know, he was really something. Right. Now, after that, I then, you know, that was the start of my academic career. But the point of telling you about these two people is that Sven was the archetypal academic. You know, you could not think of a more, anyone who would be more the archetype of an academic than Sven. He never worked in academia in his life. Bengt, he was the archetypal businessman, entrepreneur. He always worked in academia. And the point of this is that I think these two worlds are a lot less different than one might think. And especially for me, what do I do now? I tinker. I solve problems. That's what I do now in academia. It just happened. I just happened to accidentally drop into academia. I do much the same thing as I used to do in industry. There are differences. In industry, you can collaborate locally with the people there very easily. You can't really collaborate globally because you're collaborating with your competitors, and you won't really want to do that. In academia, it's easier to collaborate globally. In industry, it might be a little bit annoying because things like this happens. You know, your company gets sold, and suddenly your baby is no longer yours. In academia, that wouldn't happen necessarily so in the same way. So there are small differences, but on the whole, the similarities are much greater than the differences. And I do think that we need, in academia, we need more people coming from industry, because you can really see how you can benefit from learning from that environment before going into academia. I'm just going to finish off by saying that, you know, we have in our center, we have someone who's just submitted his PhD thesis. It's an absolutely brilliant PhD thesis. He's done fantastically all the way through, and he came to us from industry. And I do honestly believe that his path coming from industry and academia is a big factor why he's written such a fantastic thesis as he has done. And that was pretty much what I wanted to tell you about my path to academia.
Thank you so much, Jesper. I now know that when I need to make a point, I need to go and bring the toilet brush. I can see that in my head. So
well, don't forget that the toilet brush was relevant.
I know it was relevant, but even if it's not relevant, it's very funny. Anyway, so yeah, thank you so much. And as Jesper already said, he plays a crucial role in our research centers developing methods to analyze our brain imaging data into ever more sophisticated ways. So Jesper, we do have a question for you. What do you think the skills are that a scientist has that are most valuable to industry and vice versa?
Well, let's go vice versa first.
Okay.
I think that, you know, what you have in industry is very much a focus on things that work. You know, you want to develop things that work, and you want to develop them relatively quickly. And I think that's something that sometimes is lacking a little bit in academia. We tend, I think, sometimes to value novelty a little bit too much, and sort of robustness and things that work not enough. I think the skills that you can have from academia that you can take with you into industry, you know, there are things like, yes, the scientific method, just the way of evaluating how things work, and the importance of using statistics to really show that things work. Because at least from my experience in industry, I mean, we weren't good enough on that, definitely.
All right, and you said that you use lots of like maths and to do your work. What does your day-to-day look like?
My day-to-day, the best days, I mean, sometimes it's just about answering lots of emails, and those are bad days. But my best days, that's when I can sit down and actually do coding. code up the ideas I have as computer programs and test them on the data and see if they work or not. Once it works, then as an academic you then have to write a paper. But unfortunately, that's sort of the more boring part. The fun part is seeing if it works and making it work.
Cool. And so there's another question on the chat that is that somebody asking, what do you think that someone who would like to become a neuroscientist should think like?
Ooh, I think I would like to actually pass that one back to someone who actually is a neuroscientist. As I said, I'm not really, so I don't know. And yeah, I think I've thought about this as well, because I do know people who are neuroscientists, and it's a big difference because my work is all about identifying problems with the data or with the way we acquire the data or the way we analyze the data, and then solve those problems. So it's very much a reaction to something. What some people I know are able to do is actually come up with more or less completely new fields of research, completely new avenues of investigating the brain. I'm incredibly impressed and in awe of these people, because it is not something that I could ever do. Because I react to problems, and I try to solve problems, which is a different skill, I think.
Yeah, and I completely agree. Like, if you look around our centre and all of the people in it who do neuroscience, and they all, many of them are so different and have come with such different ways of thinking. And I think together that actually really makes for a very strong centre, because you fill each other in, basically, I think. I hope that answers your question. All right, well, thank you so much, Jesper. I hope that was good. That was as good for you as it was for us. And now we move on to our next storyteller, whose interest in science, and especially biology, was sparked by a rather startling discovery. Here's Celine Lee talking about her way into science. Celine?
Hey, thanks for the introduction, Carrie. Yeah, hi everyone. My name is Celine. I'm currently a PhD student at Oxford University studying clinical neurosciences. Today, I would just like to share my stories of how I became a scientist and give you a glimpse of the research that I'm doing now. So going back many years, my passion for science came from the environment I grew up in. Does anyone like K-pop or BTS? Yes, I am from South Korea, and I spent most of my school years in a very small island called Jeju Island. It's a super rural area, and my school was surrounded by big forests. I remember there were a lot of bugs in my accommodation. And now I'm gonna say something that could be somewhat disgusting, so I'm just giving you a small warning. One day I found a huge centipede crawling in my room. Not lying, it was like this big, and it had like 100 legs. So I didn't even have time to think, and I just grabbed the biggest and the thickest textbook I could see. I remember it was a IB biology textbook, and I just grabbed the textbook and I threw the textbook on top of the centipede. It made this disgusting, crushing sound, and I was covering my eyes. And I turned the textbook around, and I was expecting to see a splash of red blood all over the textbook. But can anyone guess what the color of the blood was? It was actually green. At that time, I was so shocked, so I immediately Googled why. Well, I found out that human has iron in our blood, so that makes our blood red. However, centipede has copper in its blood, so that makes its blood green. So it was very interesting how the things we observe in nature can be explained by these mechanisms at a very small scale that we can't observe with our own eyes. So naturally, I got interested in biology and went off to university in London at Imperial College to study biochemistry. Looking back, it's funny and fascinating how my journey to become a scientist started from something so small, just a centipede, and I felt that something small actually has power to make big changes in the future. So going back to my university life, when I was entering the university, I expected to learn more about the mechanisms that make up nature and human. I did learn a lot about these things, but throughout my university years and after graduating, I realized how much we still don't know about these mechanisms. So I wanted to know more about these areas of unknown and discover something unprecedented by doing research. But at that time, I wasn't sure what kind of research I wanted to do. In my final year of university, I had a fantastic chance to select the lectures I wanted to take. And I think this really helped me to learn about various areas in the scientific field and really decide on what kind of research I wanted to do. So once I took a lecture on bacteria and virus. As you might know, bacteria and virus are outside invaders that attack the human system. And the human system has a clever way to attack against these invaders. So there's this fight going on between them. And what really interested me was the ways that bacteria and virus use to hide or even escape the attacks the human system make. And I also took a lecture on cancer. I realized that cancer cells are quite different to bacteria and virus because cancer cells are your own cells that become harmful. But similar to bacteria and virus, they start to cause harm to your body and causes disease and spreads. So I think this was really the starting point that shaped my interest to diseases that arise within the human system. And more specifically, I got interested in the neuroscience and the diseases in the brain. And it was this book that really influenced me. It's called When Breath Becomes Air by Paul Kalanithi. Paul Kalanithi was a very famous brain surgeon, and his book made me think about what truly makes an individual, and are we just machineries controlled by our brain? And I realized it's the different memories and the experiences that everyone has, and the ideas we grow by communicating with others that shapes an individual. And if you think about it, with diseases in the brain, you lose all these features that make an individual. So in the end, you're losing an individual, which is devastating. Also, with Parkinson's disease, dementia also comes along where the patient loses memory. It becomes difficult to communicate with others, and the patient loses ability to control their movement. So in the end, the disease affects individuality and an individual as a whole. This is what really led me to study neuroscience, and especially the neurodegenerative disease specifically. I believe that finding a cure for these diseases is very important, but at the same time, I think finding a way for early detection of these diseases is equally important. For neurodegenerative diseases, the disease actually starts to progress years or even decades before an individual starts to show symptoms. So for example, when an individual started to show symptoms, goes to the hospital and gets diagnosed, sometimes the disease might have progressed too far and the treatment won't be as highly effective. Also, for the case of Parkinson's disease, about 20% of Parkinson's disease are misdiagnosed. So I wanted to do research on early and more specific and sensitive diagnosis of Parkinson's disease. So I've looked at many different labs across the UK to do my PhD on this topic, and luckily I found a lab in Oxford that accepted me to do a PhD project. And one of my PhD project is looking at easier and less painful ways of diagnosing Parkinson's disease. Now, we diagnose Parkinson's disease by taking a slice of brain and looking at it under the microscope. But as you might have guessed, we can only do this with patients who have passed away. Now, the scientists are developing new ways to diagnose the living patients by looking at something called cerebrospinal fluid. We call it CSF for short. CSF is a biological fluid that is found in the brain and in the spinal cord. We take the spinal cord, the CSF, it's similar to taking blood, but we use a procedure called lumbar puncture. Lumbar puncture is a very painful procedure where the clinician takes a long and very thick needle down to the lower back of your spine, somewhere opposite to your belly button. As you might imagine, it's a very painful and an invasive process. So I raised the question, can we use other biological fluid other than CSF to diagnose Parkinson's disease so it becomes less painful for the patients? So for my project, I am aiming to test saliva. I am testing saliva because for Parkinson's disease, this disease is associated with a specific protein called alpha-synuclein. This protein is found in everyone. It's found all over our body, and it is also found in the brain. It has its specific function in our brain, and for some reason in Parkinson's disease, this protein starts to misbehave and cause harm to our brain cells. And what was really interesting was that this abnormal protein was found in the saliva of Parkinson's disease patients. So I want to test if we can detect these abnormal proteins in Parkinson's disease patients' saliva and see if we can use this to actually diagnose the Parkinson's disease patients. Hopefully the test will work, fingers crossed. And just to finish up, I've always been interested in doing research that could help the patient and the clinic. I did mention in the beginning, I did say that small things can have the power to make big changes in life. And I think what I'm doing now, the research I'm doing now is small, but I do hope that in the future it will make significant changes and help out the clinic and the patients. And just the thought of this really drives me to do research every day. And that's all from me. Thanks, everyone.
Thank you so much, Celine. That's really interesting. Celine's work currently, even though she says it is small, it could have the potential to diagnose Parkinson's really easily and early on, and that could really change the lives of numerous patients with Parkinson's disease. So that would be huge. So there is a question here related to that. Someone is asking, Susanna is asking, how far can someone go with Parkinson's disease without getting any symptoms?
Ooh, I think that's different for everyone, different between different patients, but I think someone could get symptoms as early as their 30s, I've heard, and some people get their symptoms very late in their 50s or even 60s. So I think that's really variable between different people.
Okay, And there's another question. Oh, where's it gone? Oh yeah, there it is. Do you feel that there is a really large bench to bedside approach in medicine? So are the clinical impacts of the research that you are doing clear to see, and how far do we need, how far do you still have to go before this can be actually used in a clinic?
Yes, as people who are not actually working in the bench side, it's really hard to see really how far we're there to actually get like a cure or even a clear diagnosis. And as a scientist working on the bench, I do see a lot of clinical studies and clinical trials happening throughout the years. It's happening now, and it's also planned to happen for the next couple of years and decades. But there are actually a lot of clinical trials that are failing, and there are also clinical trials that some actually move on to the next stage. And actually looking at it from this clinical side, I think it really does take a long time to actually reach the patients, especially for the neurodegenerative diseases, I believe.
Okay, and do you think there's any way to speed that up?
I think to speed up, I think one of the most effective ways is to recruit as many participants as possible, and we are also aiming to recruit. Scientists are aiming to recruit patients who have just in their early stages of the disease progression, and I think that's what's really important about this early detection for the research I'm doing right now.
That's great, thank you. And so there's another question from Alison, who's asking, is this sort of research also, are your findings also potentially applicable to diagnose dementia? Other dementias.
Yes, yeah, I do think it will help to diagnose other dementia like Alzheimer's disease. There's a lot of overlap between Parkinson's disease and Alzheimer's disease, and I actually got this idea of using saliva to diagnose Parkinson's disease from a research done from the Alzheimer's field. So I think the research done with Parkinson's disease can definitely be applicable to Alzheimer's disease.
Okay, and there's another question from Venice, who is asking, what made you choose to investigate Parkinson's rather than other diseases like Alzheimer's or dementia? And do you think there is enough research for the last ones? Enough research?
Yes. Yeah, for me, to be honest, I was open to do research on either Alzheimer's, Parkinson's, or other neurodegenerative diseases, but while I was doing some research on what kind of PhD project I wanted to do, the PhD project I wanted to do was on kind of early detection and developing method for diagnosing the disease. And I think I was lucky to find this research group in Oxford, and the group I am associated with now. And this lab happened to do this research in Parkinson's disease, and that kind of got me into Parkinson's disease specifically. But looking back, I definitely could have got into Alzheimer's disease because everything's all equally very interesting.
Okay, thank you. And one last one, as it's a follow-up question to your question, the answer you gave to the bedside effect. This person is asking what your main motivation is to keep doing your research if there is such a distance between just seeing the effects put into place in the clinic.
Yes, I do agree. I am aware it takes a long time, and yeah, it's kind of difficult to it will be I mean, like, it's difficult to imagine how my research might actually affect the clinic and patients in the future, and it's kind of hard to see now. But I think you just have to be persistent and just hang in there and just keep doing research. And yeah, I don't know. To be honest, I can't see how. I can't. I think it's for us all, saying for all the scientists. I can't see how it will. I can't predict how it will affect the patient and clinic in the future, but just the thought that it will actually help in the future and it will contribute somehow, I think that really drives me to do research.
Brilliant. Thank you so much, Celine. That was super interesting. Yeah. I think it's time to move on to our next speaker. Yeah. So sometimes, for whatever reason, we don't see the opportunities that are in front of us. And our last storyteller this evening studies how we make decisions, something that she says, by her own admission, that she's not very good at. Here's Marianne Noonan telling her NeuroTale.
Hello, thank you, Corinne. It's been a really interesting, lovely evening so far. So I hope we'll end as we began. So I'm a cognitive neuroscientist and I work in the Department of Experimental Psychology. And I spend my days, as Corinne said, thinking and investigating how people make decisions and how the brain helps that to happen. Unfortunately, I'm not very good at making decisions. And just because I can tell you where in the brain is weighing up the pros and cons of the decisions that you're making here, and which bit of the brain helps you learn from the consequences of your choices here, doesn't mean that I'm actually very good at making decisions. In fact, don't you often feel that sometimes people get interested in things that they're not actually very good at themselves? So my story of how I got interested in in how people make decisions actually pivots around a decision that I didn't really make myself. Somebody else made it for me. I was 20. It was July, and I was doing a summer internship at the University of Cambridge in a brain sciences lab. I just finished my undergraduate degree in the University of York studying psychology, and I decided that I wanted to do a PhD to study this time how the brain helps you focus your attention on your goals. That was what I wanted to study. I've no idea how I actually came to that decision. That memory has just disappeared into the ether, because doing a PhD doesn't really have a clear or sensible vocation. I come from a large family in Derby, mostly made up of nurses and teachers, but my dad had instilled in me a love of science through kitchen experiments, bottle rockets that we aimed at the stars, and homemade bedroom alarm systems. I did have a lot of brothers and sisters, and I had to keep them out of my bedroom. So here I was, years later, after the last bottle rocket was launched in our back garden, shooting my shot for a career in science. Unfortunately, the current spanner in my plan was that I'd failed to get a PhD at Cambridge to study that. Not to worry, I thought, I have a plan. I'm young yet, or I was. I will go and do a master's and I'll try again the following year. Earlier that year, I had also applied for and been rejected from a PhD programme in Oxford. But that rejection hadn't actually stung as much as the Cambridge one, partly because the interviewer for the Oxford position had scared me so much, the back of my knees had started to sweat. I didn't know that the back of your knees could sweat with fear, but they can. And besides, I didn't know anyone in Oxford who I wanted to work with as much as I wanted to work with this particular chap in Cambridge. But then, out of the blue, I got a call from Oxford to say that someone had dropped out of the PhD programme there and that they had funding for me if I wanted it. I had to think. My gut reaction was no, I had a plan. My plan was to try again for Cambridge. I was so focused on that plan that everything weighing in the favour of Oxford, the funding, the security, the equal prestige, the fact that dark blue actually did suit me better than pale blue, paled into insignificance. I agonised over this decision for what felt like weeks, but was actually probably just a couple of days, until one day I happened to bump into Professor John Duncan. John is a world-leading expert in attention. He's super smart and very respected. We bumped into each other one morning in a little grey corridor in one of my favourite buildings in all of Cambridge. Where John works, and where I wanted to do my PhD, is a scientific department that's housed in just that, a house, a large but unassuming Edwardian family home. But open the door to that large, unassuming Edwardian home on a normal, unassuming suburban road, and you find some of the greatest scientists and world-leading laboratory equipment, with a lovely garden, complete with a state-of-the-art brain scanner where the garden shed should have been. But back to that moment in the corridor. I was, admittedly, a little nervous to waylay John and ask him what I should do, ask for his advice, given how senior he was. But I did. And what did he say? Well, in no uncertain terms, and in words that I've never forgotten in the last 15 years, he said to me, "Marianne, I have no sympathy for your predicament." Quite right, too. Go to Oxford, have a look at Matthew Rushworth's work. I think you'll like what he does. His advice was so forthright and so sensible that it completely knocked me off my feet as I stood in that little grey corridor in one of my favourite buildings in all the world. I started my PhD programme in the October. I met Matthew Rushworth in November, and I never looked back. That's not true. It's far too cliché. I often look back to that moment when my career is going well, or even better, when my career is going badly. And I think it was the best decision I never made. How it involved a little bit of luck, how it threw me off a path and made me flexible and adaptive to my changing environment, something that we actually need in buckets in our world right now. But that's the funny thing when you're a social animal like humans are. Social influence is so important. So important, in fact, that there are specialized circuits in the brain that help you process social information, that help you learn from the behavior of others or predict how your behavior will affect somebody else. And in our current environment, in the current situation we're in, that's so important to be able to know how our behavior might affect somebody else, keeping them safe or putting them at risk. And even more interesting, teenagers and adolescents are, in their brains, the social decision circuits are really buzzing. Social influence, particularly from their peers, is incredibly important and really influential in their decision making. And you can see that when you put a young person in a functional brain imaging scanner and you ask them to make decisions either for themselves or while getting advice from their peer or their mother, for example. Teenagers at the moment are living in a really uncertain world. And it must be so daunting for them to look ahead and think that there are these big decisions that have huge consequences in their lives. But my story, and I'm confident that of other people's as well, actually tell us that it's sometimes the decisions that you never really feel that you made that actually can lead to really unexpected and wonderful adventures. And that's where I am now. Now I'm studying how teenagers and adolescents make decisions and how they learn from the consequences of their choices. but that's another story.
Thank you so much, Marianne. So Marianne studies, as she already said, the many changes that happen in the brain of teenagers and young adults and the influence that has on their decision-making process. I think we've just about time for 1 or 2 questions. So Marianne, what are the methods that you study to look at what the impact is of those changes?
Well, in cognitive neuroscience, we have so many tools to use, and that have their advantages and disadvantages. But some of the typical tools of our trade are asking people to come in to the lab normally, and we sit them down and they play sort of very simple decision-making tasks that we often try to gamify to make them interesting, and we'll look at their choice behavior or their accuracy and sometimes the reaction time and see if we can predict their choices based on what they've done and made decisions in the past and so on. We go from sort of the simple scale experiments, we can go large scale behavioral experiments where we can try and recruit lots of people online doing these behavioral experiments. That's easier in a pandemic to some extent, but what's much more challenging generally and in the context of a pandemic is to scan people with brain imaging techniques, so we can look at sort of activity that goes on the fly in their brain as they're making these decisions, or we can look at the sort of structural white matter and grey matter, the connections between areas and the areas themselves that are really humming and buzzing away making those decisions, and we can predict how the brain and the behavior sort of come together in those, and how it changes, importantly, across the age span.
Great, and there's one more, quite a nice question to end on, I think. What is the strangest thing about the teenager's brain? Susanna is asking.
I think we've all got strange things going on in our brains. I think, well, from my personal perspective, I think it's just how things aren't always what you see, what seems to be. I think we expect far more differences than we'll probably see. And even though I could say, this bit or that bit of the brain are actually not yet mature, that doesn't mean necessarily that because it's immature, they're making bad decisions. They're actually making decisions most often in the best possible way for their environment. The adolescent and teenage time of life is such a niche period that we've all had to get through, and it's so changeable and volatile. And actually it might be the case that the brain circuits that are online and working during that period, which might be different from when you're an adult, are different to help you get through that. And then it's actually the experience of going through that period of uncertainty and changeable and learning from peers and so on that actually makes you the adult that can then exploit the new environment that you find yourself in. So it's not necessarily something that's strange, but I think it's unique and beautiful and super smart of evolution to set up this kind of really amazing plastic period of the brain. Saying that, unfortunately, that's when we know that a lot of mental health conditions come online and manifest during this period because of its changeability and so many other factors that we're yet sort of teasing apart as scientists and clinicians. So that's part of this big picture that I try and work within.
Thank you so much, Marianne, and many thanks again to all of our storytellers. What we really loved about all of these stories is how different people's career paths can be into science, and we hope you found out about that too today. So if you enjoyed today's show, do please come again and join us next week at the same time for more NeuroTales. And I'm going to now hand back to Dane.
Thanks so much, everyone, for that. Really interesting to find out what was the motivating kind of inspiration for being involved in research pathways today. I mean, there are so many ways of making a contribution to science, and everyone has their own unusual, quirky, unsuspected, and sometimes frightening kind of entrance point, whether or not that involves having green blood squashed out of a centipede, or some things that feel outside of our control. Really interesting. So thanks very much for that. Think more about various science and ideas that are happening in and around Oxford and across the world for those who are visiting from afar. Thanks for joining us this week, and we'll see you next time, same place, same time next week. Thanks, everybody. Good evening.